You may not realize it, but every day your immune system protects you from invasion from all sorts of different kinds of pathogens. Pathogens are any living organism that could get inside your body and cause harm to your living tissues. These could include viruses, bacteria, fungi, and any other living organism that could hurt you. First, there are physical barriers that normally keep out bacteria from entering into your tissues, but occasionally these are breached, like here that's depicted a cut. So once there is a breach of this anatomical barrier, now the pathogen is able to enter into the tissues. This is the beginning of the invasion. There are several layers and waves of responding cells that your immune system uses to protect you. The first are the first responding cells here, and they come in once this tissue has been opened up to the outside. And those include phagocytic cells. Phagocytic cells are cells that are able to engulf whole bacteria and viruses and be able to digest them and destroy them before they're able to multiply and cause problems. Another thing that your body uses to keep the pathogen at bay until the rest of your immune system can come in to help is a system of proteins called complement. And these are 21 different proteins that circulate through your blood in an inactive state, and only when they attach to a bacteria, they all assemble to make a hole in the bacterial wall. This releases all the insides of the bacteria and destroys it. So this system is called complement because it complements or helps out the rest of the immune defenses. That's depicted here. You can see all the cascade of proteins that have now assembled on a bacteria and have been able to blow it open, make a hole in it, and lyse the bacteria. Here there are phagocytic cells that have engulfed a virus, and here are bacteria. So those are the first responders. They hold the invaders at bay to give time for the more specific and more sophisticated counterattack that your immune system will mount. So while the first responders are holding them at bay, another very specialized immune cell called a dendritic cell picks up parts of the pathogen, captures them, and brings them back for analysis in order to mount the best immune response. So here are dendritic cells picking up bacteria and virus, and then they migrate along the lymph drainage. So these are lymph vessels that drain all of your tissues. Constantly, liquid is squeezed out from your blood, goes through the tissues like a sponge, and then drains into the lymph, leaving the red blood cells behind so it's no longer red. So this clear liquid is called lymph. So the dendritic cells use this lymph almost like a highway to flow back and take pieces of that invader back to the command and control center, the lymph node, in order to organize a counterattack. So now we're going to move to the lymph node along with the dendritic cell here. I also should tell you that when you have an infection, your body grows a lot more lymph vessels that enable you to more efficiently capture the invaders and bring them in for analysis. So moving into the lymph node, this is really the centerpiece, the command and the control center of the immune response. Inside the lymph node, and you have lymph nodes distributed throughout your body, and you may have actually felt them swell during a cold infection in your neck. They're small and they're filled with lymphocytes. Those are really the central cell of the immune system, and they enable you to make a very specific response to the particular type of invader that has come inside you. You have two different kinds of lymphocytes. There are B cells, B lymphocytes, and those are depicted in yellow up here. And you have tons of them, millions of them in each lymph node. The same with T cells, millions of T cells. These T cells each have a specific receptor that can only recognize one kind of invader. And when they were generated, back in the bone marrow, when you were developing, they were generated randomly. So you were able to recognize almost any kind of invader. This B cell can recognize a different invader than this B cell, and so on. The problem is being able to find that one B cell and one T cell that will recognize the specific invader that has come inside of you and be able to respond to it. The dendritic cells are highly specialized to do this. They move throughout the lymph node, and they're particularly situated to be able to expose the little pieces of the invader to as many different B cells and T cells until they find the perfect match. In particular, they function to present to T cells. So once they find the correct T cell, they're able to stay with that T cell and activate it. Once the T cell is activated, it expands and multiplies and produces a whole cloned army that are exactly like that one particular T cell and have that same specific receptor that's perfect to attack, recognize and attack that invader. The B cells are a little different in that they don't need the dendritic cells to present those pieces of the pathogen to them. They sit and wait, and little pieces of the pathogen can actually flow through the lymph vessel just in solution, and they can bind to the receptor that's on the surface of the B cell. The receptor that's on the surface of the B cell is also different than the T cell receptor in that it's called an antibody. It binds directly to the pathogen, and it can be released once the B cell is activated. So it's kind of like a long-range missile. Once these antibodies are released, they can go back into the bloodstream and go back to the site of infection in order to attack the pathogen. The B cell itself can sit here and secrete lots of antibody in the lymph node and doesn't even need to leave and go to the attack site. So that whole expansion step I told you about is called clonal expansion. So after activation of the correct T cell and B cell, there's clonal expansion, and then there's differentiation. So after you have a large enough army, now you need the right kind of attack to be mounted. Certain kinds of invaders, for example, protozoa or parasites, need one kind of a T cell response, and bacteria and viruses need a different kind. The dendritic cell took the right piece to the right T cell and told it what kind of a response to make. So the correct T cell is generated and in large numbers. Now we need to go back to the battle site. So how is the T cell going to know where that infection was? It was sitting here in the lymph node. It got activated. It has the right specificity, but it needs to get to the battle zone. There are two ways that we know that this happens, but we don't really understand it completely. One is that the dendritic cell, depending on where it came from, where it took up the little piece of the pathogen, it's able to imprint that information in the T cell. So if a dendritic cell came from the skin, it tells the T cell, now you can home to the skin. And now the T cell will put out the right kind of molecules on its surface that will enable it to get right to the skin. There is another way in which the T cells know how to get to the right battle zone. And I'm going to tell you about that once we step over to the battle zone itself. So after the pathogen came into the lymph node and we selected the right responding lymphocyte to attack, then they underwent clonal expansion and the T cells differentiated into just the right attack cell. We have large specific army. Now it needs to get to the right spot. So now the T cells leave the lymph node, go into the bloodstream, and now they flow through the bloodstream back to the site of infection. So here we've repeated the same drawing of what happened in the original cut. So I told you that the T cells were imprinted by the dendritic cells what major tissue to go back to, for example, the skin here. But the other way the T cells know exactly where to go, where the infection is that during the infection, all of this tissue damage that occurred released two things. One was chemoattractant proteins that give the very fine-tuned signals about where the attack is happening. And the other is that when tissues are broken, the cells that line the blood vessel, endothelial cells, express particular molecules on their surface that grab the T cells as they're flowing through the bloodstream and tell them this is the spot to exit, pretty much like an exit sign on a freeway. So the T cells are flying through the bloodstream at very high speed, and as they come along and these little molecules are on the surface near the site of infection, they get stuck on there. And they begin to slowly roll and then eventually firmly attach. Once they're firmly attached, they actually squeeze through the space between the cells that line the blood vessel wall and into the tissues to mount the counterattack. Then they follow that scent of those chemoattractant proteins to get to the specific area where all these bacteria and viruses are multiplying. So those first responders were able to hold, hopefully, the virus or bacteria at bay, keep it from making too many and getting into the blood and getting into the rest of the body. They also assisted in making those chemoattractant proteins. But now they're going to sit back and help and watch the T cells work. So there are two kinds of T cells that are in the counterattack. One is the killer T cell. It goes and scans all of your normal host cells and sees if any of them are infected by viruses or intracellular bacteria. So each cell on a regular basis displays on its surface little pieces of everything that's inside. And when the killer T cell finds a piece of a pathogen, then it immediately, there's nothing that can be done to save that cell, but it destroys the cell. So killer T cells will destroy virus-infected cells and cells that are infected with intracellular bacteria. The other kind of T cell that is in the counterattack is a helper T cell. And it's named that because when it gets activated by the pathogen, it secretes proteins that will help the killer T cell to kill more efficiently. It will also help macrophages, some of those phagocytic cells in the first responders, to kill anything they've engulfed. So it is truly a helper cell in that it organizes the rest of the immune system. Very important cell. And we can see that in the case of HIV. HIV infects a helper T cell. And once it destroys this cell, the rest of the immune system is very weak in its responses. So I told you about the cytotoxic and helper T cells. I told you about the adhesion receptors that the T cells use to get into the site. The T cells have mounted their counterattack. They've knocked down the numbers of the invader. And now we have this huge army there, using up a lot of valuable resources that you need to survive. What do we do with this army? Fortunately, they're just cells. And the army, once they no longer have any invading pathogen to activate them, they pretty much starve to death and die. So now you have all the army that you don't need anymore that has died, and now you need to resolve this inflammation and heal. So phagocytic cells that were in the first responders have another function, not just to hold the invaders at bay, but after the battle has finished and you have all sorts of cell pieces and debris lying around, things that could cause a lot of inflammation, they come along, clean all that up. And then once they clean it up, they secrete anti-inflammatory proteins that tell the rest of your immune system everything's good. There's no need to continue coming into this site. Let's get rid of those adhesion receptors on the blood vessel wall that would call in more T cells. Things are healing right now. And that allows the rest of your skin to heal in the wound healing process. That's not the last thing that happens in an immune response, though. The most important is that now, if you're ever exposed to that same pathogen again, you will respond much more rapidly, and you probably won't even have any symptoms. And the reason that happens is because I told you that most of the immune army that was generated dies. A few of them do not. And they differentiate and change into what's called memory T cells or memory B cells. So these are cells that have been activated before and are waiting for the same pathogen to arrive, and they'll respond much more rapidly and efficiently in the future. This is really important, and it's been exploited in developing vaccines. So vaccines work by injecting someone with a safe form of a pathogen, a pathogen that's been killed or a piece of a pathogen that can't really grow in you, but enough to stimulate the immune system to undergo this whole immune response that I've told you about to that pathogen, enough to generate memory T or B cells. And that's saved millions of lives in the last 50 years. So the last thing I'm going to tell you about is a little more in-depth of how the immune cells function in killing the invader. So here we're going to have a zoomed-in view of what happens when an immune cell functions in order to attack a pathogen. And this is divided into 4 different types of immune cells. The first 2 I'm going to tell you about were part of that first wave of initial responders that keep the invading pathogen at bay. The first one is a neutrophil. Neutrophils are present in your blood at very high numbers. In fact, they're the first thing that goes up, and hospitals use it to diagnose that you've actually had an infection. Neutrophils are sort of the infantry of the immune system. They attack directly, and then they immediately die. They only live in your blood for a few days. The way they work is that when they sense a bacteria through some general's feature on the bacterial surface, they shoot out these nets, neutrophil extracellular traps, that are these long strands that come out from their nucleus and trap the bacteria, holding it in its place, almost like a spider would do with a fly in its web. Once the bacteria has been immobilized in this net, the neutrophil then releases packets of basically bleach and hydrogen peroxide, very reactive chemicals that will tear open the bacteria and completely destroy it. So they're very powerful cells in that first responding wave. The second cell is a macrophage. So macrophage is also part of that first responding, but they also helped in cleaning up and in the wound healing process. So they have all sorts of things they can do, but it depends on how they are activated. If they are activated by an infection, they're attack macrophages. If they are activated by cleaning up pieces of cells after a battle, they change into an anti-inflammatory macrophage. Here, this is mostly showing an attack macrophage. Macrophages that are in an attack mode are phagocytosing, or picking up pieces of bacteria and viruses. And as they pick up a whole bacteria down here, they go through several intracellular compartments. Meanwhile, the cell has prepared packets of poisons that it now fuses with that compartment that contains the bacteria, releases all the poisons in and kills the bacteria. So it's very important that that bacteria was in a compartment. If the cell released the poison inside of itself, it would kill itself. So it's able to kill this bacteria in a very self-contained manner. The second two cells were the specific lymphocytes that I told you about that reside in the lymph node and are expanded into cloned armies to go and kill the pathogen in a counterattack. So first, I mostly told you about T cells. And here there's depicted two T cells. Up here it would be the helper T cell. And when the helper T cell is activated in the attack site, this is an artistic rendering of it giving out signals to the other immune cells to boost their responses. The killer T cell goes and scans the host cells, like I told you before, for any that are infected by intracellular bacteria, bacteria would live inside a host cell, or viruses. And once it finds a host cell that is infected by a bacteria or virus, it now fuses with that cell and can kill it in two different ways. One, by forming a membrane attack complex and blowing a hole in that cell, or by triggering a protein on the surface of that cell that is basically a big self-destruct button. And the killer T cell has just the right finger to push that button. It only pushes it whenever that cell is infected. So it's a very powerful cell. It needs to be directed against only cells that are infected. And to do that, it has that specific receptor on its surface. Then the last immune cell that I'm going to tell you about here was the B lymphocyte that we talked about in the lymph node. And the thing that was very unique about the B lymphocyte was that it could recognize a pathogen without a dendritic cell presenting it to it. And once it recognized the pathogen by its receptor on the surface, it was able to release that receptor, and then that receptor was like a long-range guided missile, where it could flow back into the site and attack the pathogen. That receptor, when it's secreted, is called an antibody. And we didn't talk exactly about how the antibody kills, but once the antibody goes to the site of where the infection is and binds to the pathogen, it can kill it in 3 or 4 different ways. One, just by covering the part of the pathogen that the pathogen uses to attach to cells, so it kind of neutralizes it in that way. Another way is that on the back side of the antibody is a location where complement proteins that I told you about at the start are activated and assembled. So it increases the ability of complement to kill a bacteria by at least 1,000-fold. The other way that an antibody can help neutralize and destroy a pathogen is that once it's coated a pathogen, then now makes that pathogen much more attractive for a macrophage, like I told you about a minute ago, to phagocytose or engulf and destroy that bacteria. So antibodies are very powerful, and they come in multiple forms, and each of them differs in either the location or their ability to perform some of the functions I just told you about. For example, IgM exists in a pentamer form. And since each tail of the antibody can help to activate complement, it is the most efficient at activating complement. It's the first antibody that's secreted because complement functions so well in that first responding wave of the immune system. IgA is unique. It exists as a dimer and mostly is found on the mucosal surfaces, inside the intestine, inside the respiratory and the lung tissues. Part of the reason that is that the protein that keeps the two antibodies together also contains a component that allows your body to secrete it across the surface and to exist on the inside of the intestines and on the inside of the lungs. And it's actually the antibody that is present in the most quantities in your body because it coats all of those surfaces. And whenever you breathe in some virus or bacteria, hopefully that IgA will bind to it and keep it from getting inside of you. And then the one that is most common and is studied the most is IgG because it is present in the highest concentrations in the blood. It exists as a monomer, and it's the one that is generated latest in the immune response after your B lymphocyte has had time to really fine-tune its response. And so it binds the strongest to most pathogens. So that's a little zoom in of how the immune cells function on a microscopic level. And I hope now that you've had a little more appreciation for what keeps you alive every day and defends you against all the pathogens that you encounter.